Today let us learn about double helix structure of DNA. DNA is the genetic material, as we all know. Genetic material is the substance which controls the formation and express the traits in an organisms, and that also it can replicate and pass on from the cell to its daughter cell, from one generation to the next generation. The most conclusive evidences as in the support of DNA as a genetic material, there are various experiments was given. First, coming to the Griffith experiment. He conducted a series of experiments on bacteria, that is Streptococcus pneumonia, and concluded that there was some factor, that is heat killed virulent bacteria strain. Heat killed virulent bacterial strain that transformed the live non-virulent into virulent strain. This must be due to the heat transfer of genetic material. In 1944, there are other scientists that Avery, McCarthy and MacLeod fractionated the killed S-type bacteria into three components, that is DNA, carbohydrate and protein. They also discovered that protein digesting enzymes, that is proteases, and RNA digesting enzymes, that is RNases, did not affect the transformation. So the transforming substances were not a protein and were not a RNA. Digestion with DNAs did not inhibit the transformation, suggesting that RNA caused the transformation. Thus, they concluded that DNA is the hereditary material. Okay. Next, coming to the Hershey and Chase experiment. They worked with the viruses that infect bacteria, bacteriophages, as you all know. This is a rough diagram. Bacteriophages that incorporated radioactive isotope of phosphorus 32p. It's important into phage DNA in a phage culture and sulfur 35s into proteins in a separating phage culture. These phages types were independently to infect bacterium Escherichia coli. Bacteria which were infected the viruses, this bacteria which infected the viruses that had radioactive DNA were radioactive, indicating that DNA was a genetic material that passed from viruses into the bacteria. It's just information, it's not at all depth, it's just introduction type. Next, coming to the DNA by Friedrich Mescher. That is DNA as an acidic substance that it is present in the nucleus. It was first identified by Friedrich Mescher in 1869. Okay, he named it as an nuclein. It was named as nuclein. However, due to technical limitation in isolating such a long polymer intact, the elucidation of a structure of DNA remained exclusively for a very long period of time. It was only structure of DNA remained until 1953. In 1953, James Watson and Francis Crick, based on the X-ray diffraction, that was the data produced by Maurice Wilson and Rosalind Franklin. They proposed a very simple but a famous DNA helical structure of DNA. It was given by Watson and Crick, and the X-ray diffraction was produced by Maurice Wilson and Rosalind Franklin. However, this proportion was also based on the Erwin Chargaff. It's also called as Chargaff principle or Chargaff rules. Erwin Chargaff formulated an important generalization about DNA structure. These generalizations are called as Chargaff rule. That is, DNA, that is double standard, the ratios between the adenine and thymine, guanine and cytosine are always equal. That is, the purines and pyrimidines are always in equal amount. Adenine plus guanine is equals to thymine plus cytosine. The amount of adenine is always equal to the thymine. The amount of guanine is equal to the cytosine. A is equal to T and G is equal to D. However, the amount of A plus T, that is adenine plus thymine, is not necessarily equal to G plus T, that is guanine cytosine. The base ratio A plus T by G plus C may vary from one species to another, but it is constant in a given species. Okay. Next, coming to the silent features of DNA. It is made up of two polynucleotide chains where the backbone is constituted of sugar phosphate and the bases project inside. The two chains are anti-parallel polarity. That means in one chain it runs from 3' to 5' and in another 5' to 3'. That is anti-parallel polarity. The bases of the two strands are paired through hydrogen bonds, forming the base pairs. Adenine forms two hydrogen bonds with thymine and guanine forms the three hydrogen bonds with cytosine. There is always a uniform distance between the two strands of helix. Okay, the two chains are coiled in a right-handed fashion. The pitch of the helix, here you can see, the pitch of the helix is about 3.4 nanometer. Nanometer is nothing but 1 billionth of a meter, that is 10 to the power of minus 9 meter, and here are roughly 10 base pairs in each turn. In each turn there are almost 10 base pairs. Consequently, the distance between the base pair of the helix is 0.34 nanometer. Okay. The plane of the one base pair stacks over the other double helix. This addition hydrogen bonds confirms the stability of the helical structure. Francis Crick also proposed the central dogma in molecular biology, that is, that at which it states that the genetic information flows from DNA to RNA to protein. It is a parent DNA and it is a daughter or sister. Then from DNA to RNA, it is called as transcription. From RNA to protein, it is called as translation. Formation of protein from the RNA, it is called as translation. Formation of RNA from DNA, that is called as transcription. Here mainly the mRNA forms. Here are some of the questions related to DNA. DNA as a acidic substance present in the nucleus was first identified by Dash in 1869. He named it as Dash. It was identified by Mischer and he named it as nuclein. As I have already told you, that is in 1869, Frederick Mischer identified the DNA as an acidic substance that is present in the nucleus. He identified Frederick Mischer in 1869. He named it as an nuclein. So option A is right. Which of the following pairs is incorrectly matched? Purines, adenine and guanine, pyrimidines, cytosine and uracil, nucleosides, adenosine and thymidine, DNA, basic molecule. Here you should observe the questions clearly that it is incorrectly matched. So purines, it is adenine and guanine, it is correct. Pyrimidines, cytosine and uracil, it is correct. Nucleosides, adenosine and thymidine, it is correct. And DNA, it is basic biomolecule. This is wrong option, it is incorrect. So option D is right answer. Watson and Crick in 1953 proposed DNA double helix model and won the Nobel Prize. Their model of DNA was based on first option, X-ray diffraction studies of DNA by Wilson and Franklin, Chargaff base equivalence rule, that is Chargaff rule, I have told you, Griffith transformation experiment, Wilson and Stahl's experiment. Here, X-ray diffraction studies of DNA and by Wilson and Franklin, that is correct. As I have told you about X-ray diffraction, it was DNA, the double helical structure was given by the James Watson and Francis Crick, and the X-ray diffraction rules was given by M. Wilson and R. Franklin. So it is right, and it was given by the Chargaff rule, that is A is equals to T and G is equals to T. Always it is constant. That is, so option 1 and option 2 is correct. Here it is, but it was not based upon the Griffith experiment or Wilson and Stahl's experiment. It is wrong. Okay, so option here it is B.